All right, this is for your Chapter 7 test Cambridge review. 16 questions, so let's go through. Before we begin, please remember that this is a study guide. It is not an absolute. So let's get started. So question number 1, note the different types of microscopes, what the microscopes can do, and the scientist who coined the term cells uses a microscope. Well, there is a light microscope, can magnify up to about 1,000 times. Remember, after this, the image becomes blurry. And Robert Hooke coined the term cells, and we'll be going over the other microscopes shortly. So know the 3 parts of cell theory. There are 3 parts. You need to make sure you know all of these. They do not have to be word for word, but as long as you have a general idea of what they are, it's fine. It does not have to be word for word. The smallest part of a living thing is a cell. All cells come from pre-existing cells, and all living things are made of cells. When we look at the first one, the smallest part of a living thing is a cell. That means if it's smaller than a cell, we don't consider it alive. An example of this would be the organelles inside the cell. All right, number 3, electron microscopes, know the difference between the different types. So this ties back into number 1, knowing the different types of microscopes. So there's a scanning electron microscope that creates a 3D image, and it's black and white. We also have a transmission electron microscope, and that creates a 2D image that is black and white. The specimen has to be cut very thin. I know it says thing, it should say thin. It should not be saying thing whatsoever. It should be cut very thin. The reason it creates a black and white image is because electrons do not come in color. All right, number 4, know the difference between eukaryotes and prokaryotes. Eukaryotes are cells that contain a nucleus. Animals, fungi like mushrooms, plants, don't forget they have a cell wall. That is something you'll need to know for the next unit. You don't necessarily need to know that they have a cell wall for this question, but it's good information to know. Prokaryotes do not have a nucleus. An example of that would be bacteria, and don't forget bacteria also have a cell wall. So plants and bacteria have a cell wall. That's what they have in common. That's really the only thing they have in common, because plants would fall under being a eukaryote because the cells have a nucleus, where bacteria would fall under a prokaryote because they do not contain a nucleus. All right, number 5, know the parts and what is found in the nucleus. So you have the nucleolus, which is smaller than the nucleus, and inside the nucleus, you have the chromatin and you have DNA. That is what is found inside the nucleus. All right, number 6, know what is responsible for making cells move. You can cross this one out. You don't need to know it, so you can go ahead and cross that one out. Number 7, know the path of proteins, where they start to where they eventually end up. So for this one, they go a little simplistic here. You should be thinking ribosomes to begin with, because ribosomes are all about making proteins. Moves to the ER, specifically the rough ER. It doesn't say the rough ER, but specifically the rough ER, because the rough ER contains ribosomes, and that's where the proteins are being manufactured. And then to the Golgi apparatus, and that's where they're going to be modified, sorted, and packaged for use either inside the cell or use outside the cell. All right, number 8, know which organelle is responsible for transforming the chemical energy from food into usable energy. And this is everybody's favorite. I think everybody's going to get this one. It is the mitochondria. Just on a side note, mitochondria is the singular form. Mitochondrion is the plural form. Cells can contain more than one mitochondria, so we would say the mitochondrion in the cell. Do not write powerhouse of the cell. Scientifically speaking, it is the powerhouse of the cell, but we have to know what that means. And the mitochondria is responsible for transforming the chemical energy from food into usable energy with the use of oxygen. Oxygen releases the ATP stored in the food. So make sure you remember that as well. That is not written on here, but you need to make sure you have that down. Number 9, know what the purpose of the cell membrane is. Very easy here. It regulates the movement of items in and out of the cell. It is a semi-permeable membrane. That means it allows some things in and some things out. If it was just permeable and not semi-permeable or selectively permeable, it would allow all things in and all things out. Number 10, know the difference between diffusion, facilitated diffusion, active transport, that is not abbreviated as ATP, and passive transport. A lot going on here. So let's start with passive transport. Passive transport means no energy or ATP is involved. Diffusion is an example of passive transport. It's a movement from high to low concentration by random collision of particles. Because it's a random collision of particles and they're bouncing off one another from high to low concentration, no energy is involved. We have facilitated diffusion, which is the same thing, movement of materials or particles from high to low. But the only difference between diffusion and facilitated diffusion is facilitated diffusion has an expressway, a VIP line that it can go through, and that is a protein pump or protein channel, but no energy is required. Then we move to active transport, which requires energy. An example of that would be endo or exocytosis, which we'll get involved in a little bit later. All right, number 11, moving along at a good pace here. Know how osmosis and osmotic pressure relate to water. When you hear the word osmosis or osmotic, there's one word you should be thinking of, and that's water. That's the first thing you should be thinking of, because osmosis really is diffusion, but the thing is, it deals with water. It's the same thing as diffusion, moving high to low, but it strictly deals with water. So it's the diffusion of water. Water moving from high to low concentration. Water moving into a cell will cause the cell to burst. We're talking about an animal cell because an animal cell does not contain a cell wall, which isn't very tough. The osmotic pressure that we're talking about is the amount of water entering the cell. When you have a lot of water entering the cell, there's a higher amount of osmotic pressure. There's a higher amount of water pressure. When there's water leaving the cell, the pressure goes down. There's not as much pressure. Number 12, know the following and be able to put them in order from simplest level to most complex. Cell, tissue, organ system. Done. It's already in order from simple to most complex. I went ahead and did that for you. If you have a bunch of cells working together, those cells will make a tissue. If you have several tissues working together, they make an organ. If you have several organs working together, they make an organ system. Number 13, looking at a diagram, know how to tell if an organism is a prokaryote or eukaryote. There's one thing you're looking for, and that's simply a nucleus. If there's a nucleus, it's usually going to be somewhat close to the center, maybe not the exact center, but it's going to be round, and you can usually tell if there's a nucleus if it has something smaller inside, which is a nucleolus. We can also look to see if it's a plant or animal cell. Animal cells are round. Plant cells are more rectangular, not perfectly rectangular, but more rigid, more stiff. And then bacteria, most of the time, those are going to be a rod shape. All right, number 14. Know how cell walls give support to a cell that comes in contact with fresh water. Well, a cell wall can handle a huge amount of water entering the cell, and it provides support for the cell. The cell wall is tough. And we said earlier in class and on the podcast, the reason trees grow up away from the ground withstanding gravity is because of all the cell walls. I asked Miss Herb to watch the dogs, and they are going crazy. I don't know if you can hear them or not, but they've been barking for like the last 5 minutes. All right, number 15, know the difference between exo and endocytosis. Well, they're both active transport, but it says know the difference. Very easy. Endo means in, things entering the cell. Exo means out, things leaving the cell or exiting the cell. Now, they do have something in common. A lot of times, students, when they answer this question, they just do the difference. But you need to know that they have something in common. And what they have in common is that they both use ATP. They both use energy. So that's what they have in common. And because they both use energy, they're both part of active transport. And number 16, the diagram. Know what the various organelles in a cell do. On the back of this paper is a diagram of both an animal cell and a plant cell. I've copied and pasted it from the bottom of the paper onto this. So I have exactly what you are looking at. So let's start here with an animal cell. We have ribosomes, which are all about making proteins. We have the endoplasmic reticulum, but you need to know there are two types. There's the rough endoplasmic reticulum, which has ribosomes on it, which is why it's rough. We have the smooth endoplasmic reticulum, which is about removing toxins from the body. We looked at a scenario of Alan, who drank a 6-pack of beer every night, and his liver would remove that alcohol from his system, and that is because the liver is made up of a lot of smooth ER. We have the mitochondria, which breaks down the energy in food and releases the energy stored in food into usable forms that the cell can use. Here we have the nucleus, and here's what I was talking about. In this example, it's not in the center, but it's a round shape. I also see another round shape, so I know this is a eukaryotic cell. So this is the nucleus. What is not labeled is the nucleolus, which is right here inside. We have the plasma membrane, also known as the cell membrane. We have the Golgi apparatus, which is used from the rough ER, which manufactures the proteins. The Golgi apparatus receives those proteins in the form of a vesicle or Golgi body, and then the Golgi apparatus will modify, sort, and package the proteins for use inside or outside the cell. We have the centriole, which you don't need to know for this test, but the centriole is involved in animal cell reproduction. You'll definitely see that next unit. All right, moving over to the plant cell, we have a cell wall, which we did not have in the animal cell. Immediately on the inside of a cell wall, we have the plasma membrane or the cell membrane. We have mitochondria, which we've already covered. The Golgi apparatus, which we've already covered. The endoplasmic reticulum, which we've already covered. Remember, smooth is about removing the toxins or drugs from the body. The rough ER has ribosomes on it, which makes it rough, and that deals with manufacturing of proteins. So the ER, you have to be specific about what type. You can't really categorize something as endoplasmic reticulum because there are two different functions that it does depending on if it's rough or if it's smooth. You have ribosomes, which are about manufacturing proteins. You have the nucleus, which is the control center where the chromatin DNA is. You also have the nucleolus right here in the center. In a plant cell, you have two things that you do not find in an animal cell. One is a large, giant vacuole, which is like a giant UPS warehouse. It's going to store things. Most of the time, when it stores liquid, that liquid is going to be water. It also has chloroplast, which is involved in photosynthesis. So that's going to be it for this. Make sure you take a look at everything, including the Kahoot that was sent out earlier. If you have any questions, please do not wait until the morning of your test. Please text me or leave a comment or just come see me between classes. I will be there after school tomorrow. It is early release. I have a meeting at 1, but I'll be there in the meantime. So if you need help, please feel free to swing by. All right, good luck on your test. We'll see you guys in class.